Hello, virtual listeners. We are Team Toxic Algae Blooms. I'm George Nahas, and I'm with Ben Ginkley. And we're excited to share with you the results of our work the past couple weeks regarding toxic algae blooms and frugal methods to detect them early. So without further ado.
Phytoplankton are some of the oldest and most abundant organisms on Earth. They're the photosynthetic bedrock of the ocean's ecosystems, and they produce the dominant fraction of oxygen that we breathe in the world that we live in. However, not all constituents of these diverse taxonomic clades could be considered friendly. Many species excrete chemicals that are either neuro or hepatotoxic. These toxins accumulate in the environment, primarily in small fishes and in shellfish, and they're then eaten by larger animals like mammals and birds. The poisoning can become fatal in these animals, which is how a lot of these toxins were first discovered. A harmful algae bloom coincides with a change in the phytoplankton community structure, where toxin-producing species become significantly more abundant very quickly. Given the wide array of different organisms producing toxic compounds, both in marine and in freshwater environments, we decided to narrow our focus a bit and focus specifically on the Pseudonychia genus in the Pacific environment along the California coast. Pseudonychia species are pennate diatoms. They're found all across the world. They produce domoic acid, which is a neurotoxin that causes a disease state called amnesic shellfish poisoning. ASP lethality has been documented both in marine mammals and in humans. The main problem of toxic algae blooms is that as the ocean temperatures begin to warm, this lengthens the toxigenic growing season of these species. So this is a growing problem that will get worse as years and decades go on. Therefore, scientifically, it will be critically important to, at a minimum, establish robust baseline measurements of phytoplankton community structure to effectively detect and quantify changes as they occur over the coming years and decades. Just as measurements of sea ice coverage goes back only as far as our ability to effectively measure it, so too do we need to catalog phytoplankton species, their relative abundances, to draw conclusions in the future about what changes are happening in our oceans. This led our design team to the question, can we develop a frugal method to measure and detect harmful algal blooms before they pose a public health threat?
So in the process of thinking about how to approach this problem, we considered a variety of methods. Two of them, in theory, are frugal. That is 1 and 2 here, thin layer chromatography and frugopolarimetry. And the third one, while it sacrifices some in the frugal department, it in theory has a higher degree of accuracy and sensitivity and may be able to make up for some of the weak spots, and where the other two may not be able to handle due to their low cost nature. But we will talk through the pros and cons of all 3 of them. Pros and cons, starting with thin layer chromatography on the next slide. So thin layer chromatography, TLC, is a technique used in organic chemistry to separate molecules out of a heterogeneous mixture based on polarity. TLC uses a plate, which is paper-based. Many times in practice, it is silica gel and some kind of solvent, and then depending on the molecules you're trying to separate, they will interact with the solvent depending on the solvent's polarity, and that's how they move up the TLC plate, allowing us to see what heterogeneous mixtures are made up of. That's a very 10,000-foot overview, but as it pertains to harmful algae blooms, as the main cause and the main detriment to animal and human health from these blooms comes from the neurotoxins that are released. You can almost remove the actual phytoplankton themselves and think more about the toxin. These are 2 examples of toxins found in harmful algae blooms. Domoic acid is the main one released in Pseudonychia blooms. And you can see it's a highly polar molecule. And if the polar mobile phase was used in a TLC technique, it should be able to be separated from the seawater. The reason we tabled this is because neither Ben nor myself have extensive expertise in paper-based diagnostics. And past that, some sample preparation will be required. So on our Notion page, we've outlined some of these steps that may need to be taken, namely filtration and extraction. In order to kind of purify the sample and remove any particulate matter, these steps probably would be required. But seeing as we don't have access to a lab or harmful algae blooms to test this, we just wanted to list this as a potential route for future investigators to go down in relation to this problem. But this was not the main focus. The other reason that we tabled it that may be, more relevant to its maybe potential not best practice in the field is that there is some kind of expertise required in running a TLC plate. Is absolutely not the hardest thing in the world, but there is some startup knowledge required in how to, figure out the right solvent and assemble the device properly. That if you're a citizen scientist and oceanographer, you may not have had organic chemistry, and this may not be exactly entirely in your wheelhouse. So we tabled this for that reason, but it is on the Notion page and worth exploring. Perhaps a more viable route that we spent more time thinking about is frugal fluorimetry. So fluorimetry is a technique that's widely used in many areas of science to detect the emission wavelength of electrons when they are excited by another light source. In 2013, Emmanuel Boss's group published this paper in Sensors that described a frugal fluorimeter that made use of one LED, a red filter, a convex lens, and a photodiode to detect the levels of chlorophyll A in saltwater environments. The reason they were doing this was for a similar principle to detect the levels of chlorophyll A produced by phytoplankton, and it provided a great framework for us to kind of build off of and introduce some design implementations that may expand the idea and make it more relevant to detecting pseudonychia. So we want to expand this to be a flow-through design, meaning so you have a sample reservoir that is pumped through an acrylic sample tube that is clear, and then we can have multiple LEDs driven by an Arduino microcontroller, and each LED will excite a different photosynthetic pigment. In theory, excite a different photosynthetic pigment, all three of which are found in pseudonychia. These LEDs could be turned on or off based on the Arduino microcontroller, and based on the emission wavelengths we get out, we may be able to say that there are photosynthetic pigments in the water, and this can lead us to detect harmful algae blooms sooner. There are a couple caveats with this. The one being that chlorophyll A, phycocyanin, and beta carotene, while they are primary, normally found in high amounts in pseudonychia, they are also present in other phytoplankton. These are not exclusive to pseudonychia. And from our literature review, we didn't find any photosynthetic pigments that were exclusive to pseudonychia. So in essence, you may detect these, which may be an indicator of a harmful algae bloom, but you can't necessarily say it's pseudonychia harmful algae bloom. One other design point I want to point out here that we've included on this iteration of design from Emmanuel's original work is putting in a sheet metal casing. In his original work, the sensor was designed to be used underwater. We wanted this to be able to be handled kept on a boat. So our original design is having a sheet metal casing over it that will actual electronics in the sample, not only eliminating the effect of rogue EM waves from the pump, but also making the whole apparatus dark. So this apparatus could hopefully be used in daytime or nighttime. This needs to be taken with the grain of salt that this is a very preliminary design. This has not been field tested or even lab tested. So there's a lot of future work that needs to go into it, but this is how we envision the project moving forward, and this is where we spent most of our time preparing. So Ben now will talk about maybe the big brother to this fluorimetry technique that is more sensitive and more specific, and I will pass the baton.
Right, so you can consider flow imaging microscopy to be like a big sibling to fluorometry. You get more data, more information, but it comes often at the cost of higher component expense and a higher power consumption cost. Commercial devices that are on the market now, a benchtop version called a FlowCam, or a field deployable marine environment buoy type design called the Imaging Flow Cytobot. Both of these are very sensitive and produce high quality images and can operate autonomously to varying extents, but their MSRP is over $100,000 per device. This makes them incredibly inaccessible not only to most researchers, but especially to any citizen science type initiative where you hope to decentralize the sample collection and sample analysis process. To address these gaps in funding availability specifically, academics have been working on more frugal designs. So the lab at Stanford has developed the Plankton Scope. A lab at Northeastern has developed a device called the Artemis. Both of these are much less expensive than the commercially available devices and offer similar fidelity in the image quality that you get out of these devices. However, comparing back to fluorometry, the higher resolution data you get from imaging comes at the cost of requiring greater power and greater computation to actually get usable data from the images. So whereas a fluorometer can run off of batteries and an Arduino, typically to take images like this, you need to be at least running a Raspberry Pi, if not a more sophisticated piece of computer hardware, which precludes the ability for you to run on batteries for longer than maybe a day or two. The upside to this is with imaging data, you can train computer vision algorithms to be able to automatically discern between different plankton species. And this gives you the ability to, in single images, single shots, be able to make conclusions about the phytoplankton community structure, not only establishing an effective baseline of what the community looks like on a regular day, but also giving you insight into potential changes in the community structure that would indicate that a bloom is imminent or is already happening. At the end of the day, the goal is to engage citizens around the world, whether they're in the ocean or on shore, to begin collecting data and sharing it with one another. And so citizen sciences played a central role in our design thinking throughout this process. And Georgie will talk more about that.
Yeah, as been said, we want to build something that is going to be frugal enough and easily accessible enough that can be used by the legions of citizen science oceanographers that exist out in the world. People that either sail for their job relating to research, or more exciting maybe, people that just have boats and enjoy being out in the ocean sailing. If we can harness this huge community and get involved with them, then it's possible we can really expand our solution in a massive way that would otherwise not even a lot of money could buy. So this is a mock-up of a website that we this is not a final product, but this is just kind of how we could envision engaging with the community and, encouraging open access sharing of data, as well as providing a platform for these kind of measurements and information to be shared and disseminated. So this would just be a standard homepage. But if you go to the next page, this is kind of a logical flow of how we would imagine interacting with citizen scientists. So we would have an opportunity to either build or purchase a Frugal Perimeter, or a kit to build your own Frugal Perimeter, which is more fun. And then people would take these out to the ocean. They would record their measurements like these excited scientists. And then at the end of the day, they could take their measurements and go back home and actually log it on this website. So on the next page, we've drafted up a sample of what we imagine this logging, prototype looking like. This is, there's a lot of design improvements that could be made to this, but basically if you're sailing the Pacific Ocean, you go to the Pacific Ocean tab, write down the coordinates and the conditions of where you were, and then just log what the three channels gave you, the chlorophyll A, phycocyanin, and beta carotene. And just as over time, as more people use this and data gradually accumulates, we'll be able to establish a background fluorescent reading of the ocean, which will then allow us to determine when perturbations are occurring in this fluorescent signal. And in theory, if this signal goes up, it would indicate there's a higher level of photosynthetic pigment in the ocean that we could then use to start detecting when harmful algae blooms are occurring. And again, as it stands now, the Frugal Perimeter wouldn't necessarily allow you to say that Pseudonychia is in the area, but it would allow you to say, hopefully, that there is a harmful algae bloom coming in, as there is a couple standard deviations more photosynthetic, higher levels of photosynthetic pigments in the ocean than there were the week before. So that is the dream with this project, and that's where we hope it goes, getting to engage with the masses of people that are already out there sailing around and enjoying the ocean.
So with that said, our project is very early in the design life cycle. We're pre-prototype at this stage, so there's a lot of room for us to bring on additional collaborators, whether they're to help with prototype development or to assist in developing the community to actually deploy these devices to citizen scientists who can begin collecting data and sharing it with one another. Last, we'd like to give special thanks to Emmanuel Boss and Bibi Kale, whose work a lot of our designs are directly predicated upon. So thank you to them for not only advising us, but for laying the groundwork for some of our future design work. We also like to thank Tyler and Manu for organizing and facilitating this course. We wouldn't have met all the people in the course as a whole, the people in this project. Georgie and I would not have met each other, and same goes for everyone who kind of stepped in and stepped out of this project at various times. None of this would have been possible without Frugal Science, so we owe a huge debt of gratitude to the organizers of this course. Our contact information is here on the screen now, and we also will share links to our Notion page and our Discord team. If anyone would like to get in touch, connect with us, collaborate, we're happy to chat about algae blooms, about the environment, and developing prototypes in citizen science. So with that, I'd like to thank you all for listening and watching this video. And Georgie, any last words?
I firmly second everything that was said. Thanks for everything.
Awesome. All right. Thank you everyone for watching.